Okay, I'm not quite sure whose computer we're going to do Randy next, but I'm going to take the opportunity to introduce Randy, very briefly. I first met Randy when I went out to St. Louis to visit what was a new colleague at the time, Steve Peterson. Steve, as Randy had mentioned, had been doing PET experiments within, what was then, kind of the center of the brain mapping universe, the PET imaging group at WashU. Steve was an awesome guy, a lot of fun to hang out with, but fundamentally a skeptic, and really, was not compelled by the early data that we showed him. But he said, boy, if we could, reproduce some of the early language data, if we could see, a verb generate task, whatever, then he would consider it interesting and potentially worthy of the attention of one of his grad students who was graduating at the time, Randy Buckner. So actually, he and Randy came to Boston. We did the experiments. I think in the end it didn't work, but somehow we didn't completely dissuade him that it had the potential to work. And sure enough, a short while later, Randy came to the lab, and the rest was history. Did a series of beautiful and very elegant experiments with his colleagues, including Anders Dale. They used to cost me a lot of money when they wanted to go out for sushi at night. They would usually come and grab me and invite me along, and I felt, very privileged to be taken along. And really, they were just taking me along because I picked up the bill. And they were both big guys, and they could eat a lot of sushi. But they were fun dinners and many fun times and much great science. So with that, Randy.
Well, thanks so much for having me, and I say that in the most general sense, not today, but having me in the laboratory. It really was a remarkable time in the field, and it's hard to capture the excitement given, let me just click this on, given, there you go. The excitement, it's on. The excitement at the time, because here for the first time was a way to potentially conduct experiments looking at brain circuitry over and over and over in the same individual, and potentially even faster. I think it was even more exciting to us, those of us using PET, because we were so limited. We were always up against radiation constraints, but also we were keenly aware of the sluggishness of the technique. So I don't think it's a coincidence some of the people coming from PET were some of the first individuals actually take advantage in terms of applied sense of these fast techniques, because we were the ones so burdened by the sluggishness of our existing techniques. And so I wanted to build on Roger's talk, and he talked about elegant studies that taught us about the human visual system and how we extract out things such as the identity of objects and images and the colors, and how the visual system is exquisitely organized to allow you to perceive something like this beautiful image by Van Gogh. I want to shift gears entirely and talk about the brain circuitry that allows you to create an image like this by Van Gogh. And in this sense, I'm glad that Bruce talked about the history and Jack's perspective on this, because early in 1990, I think he hit the nail on the head. He was saying that these techniques had the potential to teach us about parallel distributed circuitry across the brain, and that is exactly the kind of thing that was inaccessible, I would even say, in animal studies, to be able to fully appreciate the extraordinary architecture that goes beyond what an individual area of the brain does, but rather what can be accomplished by circuits across the brain. And I'm hoping to just give you a little bit of a flavor to that. And so I wanted to go back in history as well, but a little bit further than the other speakers, about 3 million years. And so here is just an estimate of actually something that happened to the hominin line, that is our ancestors that lead to us and not to other great apes. Over the last 3 million years, there's an extraordinary explosion that you can see in the record from fossil endocasts, and this is a plot of brain volume. And around here you can see that we're going along a linear trend from about 7 or 8 million years ago to 5 million years ago to 3 million years ago in the hominin line, where our brain is expanding. Many primate lineages have this brain expansion as if a larger brain, a bigger brain, a more capable brain is, holding all else equal, a winning strategy. But almost all other species are held back. in terms of brain expansion, and then this remarkable expansion around 2.5 million years ago. You see a tripling in brain size in the human line that leads to our niche, this incredibly capable cognitive species that we are. And we know a little bit about what happened in that time by making estimates for living species. And so it's fortuitous that the modern human brain is about triple the size of the modern great ape. And in the presence of that, you can ask the question, well, what has expanded or what's different between our species? And I just want to mention one specific finding, is that if you look at the absolute size of primary visual cortex in a chimpanzee and a human, they're about the same absolute relative size. A human's a little bit bigger, but that's in the presence of a tripling in size of our overall brain volume. What that suggests is other stuff disproportionately expanded. And what is that other stuff? And we have some insight into this from comparative studies that have been done for over 100 years now. These are maps. We often talk about Brodmann areas in our field. These are actual images produced by Corbinian Brodmann in a monkey. It's not a macaque, but it is an old world monkey. And here's an image that he produced in the human. And basically what he's doing is looking under a microscope at the tissue sections of different parts of the brain. And by looking at the laminar structure, the cellular structure, and the myelin structure of the brain, he can make estimates of homologies, that is, areas that are the same across different species. And you can see here, area 17, what is the primary visual area V1 in Roger's talk. Here it is in the human. Here it is in a monkey. And what you immediately appreciate is the disproportionate relative turf that it takes up in a monkey as compared to a human. When you go and you look at other areas, there are a number of homologous areas all throughout the visual system into parietal cortex, basically this sensory motor hierarchy that goes all the way through. Motor regions is largely homologous between the monkey and the human. But what Brodmann also noted is regions such as area 39 and 40 in the posterior parietal lobule, inferior parietal lobule, and also these areas in prefrontal cortex that he himself did not think had homologs in the monkey and the human. I actually think there are homologies, but at the very least, they're tremendously expanded in the human brain as compared to the monkey brain. And functional MRI fundamentally was a breakthrough in many ways, but one of them was that it allowed us to be able to chart these large-scale circuits across areas in the brain, and critically to do so for these regions that are expanded in humans that presumably underlie human cognitive capacities. And so how would you do an experiment like this? There's a number of ways. I'm not going to go into any of the technical details, just to suggest that they're powerful but also have many limitations and caveats that I'm not going to discuss. But basically, you could look at a portion of cortex in the human and simply ask, as Roger did in his last set of studies, what is it functionally correlated with across the brain? And it will give you a first-order estimate of the network. And to give you an example of that, here is a region in human. This is 1,000 individuals average on the cortical surface, taking advantage of these same techniques that were pioneered to do the original retinotopic studies. And if you take a region in what is area 7, and you look at what is functionally coupled to, you actually see a constellation, a network of areas that involves extrastriate visual cortex and these regions in motor cortex, including the frontal eye fields. And this is not an arbitrary constellation of regions or an arbitrary seed and network that I'm showing you. This is the canonical sensory motor hierarchy that has been well studied in monkeys and in humans. This is the network of regions, the hierarchical progression that if you're perceiving something in the outside world and acting upon it, such as picking up a cup of coffee, and I probably shouldn't prime you that. I know you've been sitting for a long time, but if you imagine you were picking up a cup of coffee, you would be using your visual system to extract information about that object and using transformations in these parietal regions and the relationships to these motor regions to actually guide that motor act. This is the canonical sensory motor hierarchy. And so what is super powerful about functional MRI is it's allowed us to get insight into these exact parallel distributed circuits that Jack talked about more than 25 years ago. And just to show you that, I just want to illustrate a little experiment that Thomas Yeo did several years ago. And what I'm going to do is I'm just going to repeat this experiment and move the seed region across these parietal association regions of cortex. And I want you to see a couple things and to watch that carefully. One thing I want to do first is just anchor you to this sort of canonical sensory motor hierarchy. And now watch how the network of regions changes as the seed moves across the cortex. The first thing that you notice as you move across these association regions, you get dramatically different network architecture. Right next to one region is another region that's embedded in an entirely different circuit. The other point I want you to take from this is that each of these networks, wherever you are, you tend to get these distributed networks of cortex. And here's another canonical network. They tend to always be distributed across frontal regions, temporal regions, and if I show you the midline as well, they sort of have this sort of global commonality. It's as if there's one grand plan that evolution found and it just duplicated and extended it. Now, the third thing I want to illustrate with this experiment is that when you look at some of these association regions in parietal cortex that are not too far from the regions that are involved in this sort of canonical sensory motor hierarchy, while they're distributed and parallel, they actually have an interesting feature. They're as far away as you can be from sensory and motor regions in the cortex. While they have that same distributed structure, they do not seem to be, or sort of anatomically at the opposite end of the cortical architecture than you would be if you were actually in a perceptual system or in a motor system. So, what are they involved with? And this distributed network is often now called the default network, but I think it's just sort of the canonical example of a higher order distributed association network. And here it is now plotted on the right, just to show you the architecture between this canonical sensory motor hierarchy and this other distributed network. So what is it involved with? One clue comes from another experiment you can do. Rather than placing seed regions and looking at the architecture going from perceptual systems outward, such as in visual cortex, you can go from mnemonic systems. So if you put a seed region in the hippocampal formation, the image to the left is what you get. These distributed associated regions that are not embedded within sensory motor hierarchies seem to be embedded within mnemonic processing streams. And right off the bat, that suggests you a feature. I think that's an important insight to a feature of their processing functions, rather than being systems and circuits that have, through evolution, have become specialized to process information from the outside world and help you act on it in the here and now. These are distributed circuits that seem to be involved in your memory systems that allow you to extract information from your past and process it, and potentially internally, devoid from the outside world. We have studied this extensively, and one idea we've come up with, and many others, Mark Rakel has talked about this, Michael Fox, as I'll mention in a minute, is it seems to be the case that there seems to be a broad distinction of processing modes. Loosely, and take this heuristically, not specifically, this canonical sensory motor hierarchy is involved in external attention. When you're taking in information from the outside world and operating on the here and now. This distributed network here that you're seeing that's involved with memory systems, as among other systems. These are very complicated circuits. This is just a heuristic presentation. For lack of a better word, seems to be involved with internal mentation. How do we know this? If you have somebody in the scanner and you ask them, think about what you did yesterday, this is the network that will become most active. If you think about what you might do tomorrow or what you might do when you get up and leave this, there's been a large number of talks. I guess one thing is that if you're drifting and you're not paying attention to me, I'm okay with that, because you're doing the exact experiment that would activate this second network if we had a scanner right now involved in internal mentation. And there's another really interesting feature of these two networks that was illustrated a few years ago, and that was by Michael Fox and Mark Rakel. It's not only that they seem to be involved in very different modes of processing. All indications are that they're competitive with one another. That when you're doing this, you're engaging these circuits involved in taking information from the outside world and operating on it, you're not in these internal modes of cognition. And when you go inside yourself and detach from what I'm saying and think to yourself about the food or the restroom that you're going to visit, you're actually not activating the external modes. And he showed this by actually showing what he referred to as them being anti-correlated with each other. And I think this is a critical insight. So not only was these functional MRI measures for the first time able to show you something about distributed networks across the cortex in a human that potentially underlie higher order cognitive function, those things that allow us to think, to remember, to plan. They also suggest that there might be fundamental battles between networks that are going on in all of us in healthy, functional ways that allow us to sit and listen to a talk, to think about what it means to us, and to plan what we're going to do, basically to extract information from the outside world and also use that and weave that with our memories to actually plan for the future. Now, just because we are running behind in a bunch of the talks, I'm just going to skip over this very briefly. But one other discovery that's been made by a number of laboratories is that not only these large-scale systems that are involved in fundamentally different modes of processing, there are also networks that, for lack of a better word, might be helping to regulate them, to control them. And one large network is sometimes what we call the frontal parietal control network, shown in yellow right here. Wedged right between. I think this is not an anatomical coincidence. Anatomically situated between these other networks, and so an important question you can ask is: are these the regions? Are these the networks that have expanded over evolution? The question that it led into, and it's very hard to estimate this. We don't actually have a record of how brains evolved. We know that there's a tripling in overall brain size over the last 2.5 million years, but what expanded? Was it proportionate? Were some of these areas, as I alluded to from the evidence from the living chimpanzee? And David Van Essen did a really nice analysis that provides some insight. It's very hard to take the details literally because they're so unconstrained in terms of what we know about evolution, but he did a very important analysis where he actually took what we knew about the modern monkey brain and took the cortical surface and took the human brain, and he was able to come up with 23 different areas that he thought we had good evidence that were homologous between the monkey and the human. And for those of you who don't know, the human brain, the cerebral cortex, is about the size of a large pizza. A macaque is much smaller, so the question becomes, if you align all of these homologous areas, primary visual cortex, primary motor cortex, certain regions along the midline, you can ask the question, well, how would I have to expand and stretch the monkey brain to get it to match the human brain? And it's not uniform. And in fact, this is a plot of the estimated expansion of the last 25 or so million years of evolution on the cortex, the disproportionate expansion, when you take these landmarks of the monkey brain. And I think it's very interesting that it is not the case that any one zone of the cortex is disproportionately expanded. For example, it's not just the prefrontal cortex. It is distributed association cortex in all of these zones, in parietal association cortex, prefrontal cortex, and the temporal lobe, that represent these interwoven networks that we know to play an important role in higher order cognition. And I'll end this by whetting your appetite. I think this is something that you'll hear from Sue Whitfield-Gabrielli more about, is I think not only does this shed insight in some of the deeper questions of that, how have brain circuits evolved? How might they underlie higher level cognitive thought? How might they allow us to be creative, to plan, to simply think about things that are not in the world around us? Sometimes I say one of the great inventions of the circuitries allow us to go from being a trial and error learner like you see in rodents to a mental explorer, which you see in all of us. I think it sets a foundation for also understanding mental illness, because some disorders of cognition, I think, are best understood as the fragile balance of these complex networks and circuitries that are in competition like we talked about. And just to whet your appetite, if you actually do mathematical models that just characterize the broadest features of how these networks interact, this network here they're referred to as being involved in internal mentation, this sort of non-canonical parallel distributed network over here, the control network that sits between, and these other networks involved in external attention. In healthy typical individuals, you get a nice architecture and segregation of these networks. When you look in individuals with psychosis, and this is something that Justin Baker showed elegantly, you see a breakdown in the organization and the separation and the integration of these networks that you'd see in a typical individual. And I think we might be getting glimpses about how large-scale circuits become dysfunctional, and I think that's something we'll hear more about from Sue Whitfield-Gabrielli. And I just wanted to end by saying how remarkable it is. I was in a laboratory, I remember seeing this image, thinking that's intriguing, that's something that might be used to study the brain. And then we saw one after another remarkable demonstration about how one could use these techniques to look at intrinsic signals, and then the work that Roger showed you and the work you'll hear from John Gabrielli. These were early papers that came out when I was still sitting and wedded to PET. And I think Bruce Odom was very generous when he talked about St. Louis, alluding to the fact that we went kicking and screaming. You have to imagine a time. We were one of only 3 laboratories in the world that could measure the brain at work. And here are now technologies that threatened that everybody could do this. And this is an important threat, because I think through the dissemination of these tools into many laboratories, in many hands, many computational and people using novel computational approaches, engineers, cognitive neuroscientists, psychiatrists, neurologists. I think it's actually through that dissemination that has led to what I think is, for me, one of the more remarkable discoveries. We now know a lot about the distributed architecture of thought. And so I'll end there. Thank you.